Okay. Uh, Before I begin, again a check to make sure you can see my slides. There is my pointer at the top of the slide. You can see it.
Yes, sir.
Yes, yes, sir.
Okay. Pointer way at the bottom.
Yes, sir.
Yes, sir.
Okay. Left
Sir,
Yes,
it's
sir.
perfect.
and right.
Yes, sir.
Okay. Great. Okay. So up until now, we have been looking uh, at DNA and its function with respect to how it gets replicated, then issues with respect to DNA and how it gets repaired. Okay, if there is damage to the DNA, and we also looked at what basically are mutations as far as DNA is concerned. And somewhere there, I don't know whether it's there in the syllabus or not, but I also covered solid phase DNA synthesis. Okay, thought that it is nice for you to know that. Okay, now we are going to look at the second aspect of the function of DNA, where its its function as the repository of what is called as the holder of genetic information is concerned, and that comes to the forefront. Okay, so in the first part we look basically at how DNA got replicated, and during replication what happened, and if there are any damages, how they got corrected. The other function of DNA, as you all know, is that it also carries a lot of information which is necessary for the everyday functioning of the cell and for the cell to respond to several stimuli. Okay, so we are going to now concentrate on that aspect, and to begin that, we are going to look at, as it says here, the transcription of DNA to RNA. Okay. Now, the origin of the word transcription is also very interesting. Okay, if I were to ask you all to do a little exercise, as you are sitting there, hopefully you're having a piece of paper in front of you or something like that, and if I ask you to simply transcribe what is there on the screen here, okay, DNA transcription to RNA, and do that on your notebook there. Okay, so I'll give you about 10 seconds to do that. So I'm asking you to just transcribe what you are seeing on the screen, DNA transcription to RNA on your notebook. And if you have done it, one of you can unmute and say, yes, I have done it. Okay,
Yes, yes, sir, done it.
done it. Okay, great. Now, I'm also going to tell you to translate it. Okay, and when I mean translate it, I want you to write what you're seeing on the screen in a language other than English. Okay, hopefully your mother tongue. If not your mother tongue, any other language you're comfortable with. Okay, and do the best you can. Maybe you don't know the word for transcription in your language, but then try and do the best. Okay, you can. Can you?
Yes, sir.
Okay. So who was this who said yes, sir?
Shivam, sir.
Okay, so Shivam. So, can you tell me about what you wrote when it asked you to translate it? In which language did you write it?
No, sir, I'm yet to write. I said yes, we will do.
Okay, just do it now. After you do, and let me know. I'll give you another maybe 15 seconds to do the second part. That is a little more difficult actually. Okay. So anyone has finished both transcription part and translation part? If you can unmute and tell me, that would be nice. Okay. Has anyone?
So it is difficult to translate that
Yeah,
word
it's
into
okay. Yeah, it's okay.
Hindi.
How the best you can do. Best you can do.
I wrote it in Hindi, but then I don't know the exact word translation of how it occurs or how
Okay,
it.
okay,
I, I,
okay.
j-
Yeah, it's okay. It's fine. It's fine. I'm not actually interested in what is the Hindi word for transcription or say Marathi word for transcription? Maybe Sakshi knows. Sakshi, did you write what is the word for transcription in Marathi?
Sir, I googled it actually.
Ah, Shanelo. Okay. Okay. So,
Prati.
anyways, so what is important is this. Okay. When I asked you to transcribe, no, if you look at what you have written, no, you have written it in English only. Okay. The only difference is you have written it in your own handwriting. Okay. So you will still find in what you have written a D which looks somewhat like this, a N which looks somewhat like this, or if you have written in lowercase, you have written as lowercase d, lowercase n, a, and so forth. But when I ask you to translate, you suddenly find that what you have written is in a totally different script. Okay, what we call typically as Devanagari script, right? So what say Shivam wrote or what Sakshi wrote would look very different than the English alphabet. So now you have got this, matra and kana and all these things as you try to write DNA transcription to RNA. Okay, and basically that is the origin of the word. So when you are doing transcribing, no, essentially the language of transcription is same as what you started with. So if you look at what DNA is composed of, it is composed of bases, sugar, and phosphate. And when it gets transcribed, it is also converted to RNA, which technically speaking is also composed of bases, sugar, and phosphate. The difference is that this has a ribose sugar and this has a deoxyribose sugar. And maybe in this case, instead of what do you call T, you have a U. Okay, this is ATGC and this is AUGC. But as far as the chemistry is concerned, both are chemically kind of the same entities, what we call as nucleic acids. And because you are going from one type of nucleic acid to another type of nucleic acid, we call it transcription because the chemical language is kind of the same. But the minute you take RNA and convert it to a protein, now the language is that of amino acids. Okay, because now instead of nucleic acids coming together to give you a polymer, you have amino acids coming to give you a protein. And therefore you have actually changed this, in a sense, the script. And that is why conversion of RNA to protein is called translation, because you have now gone away from the language of nucleic acids and gone into the language of what are called as amino acids. Just like you see on your notebook, when I asked you to transcribe, it looks exactly like this, except maybe your handwriting is different or you use uppercase and lowercase. But when I told you to translate, suddenly it is in a very different script, although it is giving you the information which is contained in the sentence, but in a different fashion. Okay, and that's why we use the word transcription and translation to denote the conversion of DNA to RNA and then RNA to protein, which we call is the flow of genetic information. Okay, so in the last few lectures we were looking at this part. Remember, replication, mutation, repair, recombination, and solid phase synthesis. Essentially, that is DNA to DNA, DNA to DNA in a sense. Now we are going away from DNA to RNA and then RNA to protein. But since RNA chemistry wise is very similar to DNA, we call this process as a transcribing process, and then going from RNA to protein as a translation process. Okay, so this is what we call as the the central dogma, as it is called. Okay, now most biological systems have information flow which occurs from DNA to RNA and via RNA to proteins. So the information that is coded for in the DNA, the bulk of it is in dictating what protein will be made, which is necessary for the life functions of the cell and therefore the life functions of the tissue or then of the organism. But in addition to this, we know that in nature there are some entities which have RNA as a genetic material, especially things like retroviruses, where RNA gets converted initially to DNA and then from there the information flow can occur in the same fashion. Okay, so with this as the basis, what we are going to look at in the next, in this lecture and maybe the next lecture, is this process of how DNA gets converted to RNA in its journey to be later translated to give you the protein. Okay. So, what is the definition of transcription? Okay. The cellular process in which RNA is synthesized using DNA as a template is called as transcription. Okay. So this RNA that we are going to talk about is actually related to the DNA in that this DNA is being read as a template, and using the DNA as a template we make the RNA. This process is very similar to what happens during replication, because in replication, if you remember, each of these strands separate and then each of them acts as a template to make what is called as a daughter strand, okay, or the replicated strand. Something similar happens here, except the fact that when one of the strands of DNA is read as a template, what you get from reading that as a template is an entity which is chemically similar but not identical, and that is RNA. Okay, so that process is referred to as transcription. Okay. Now, the difference between what we call as replication vis-a-vis what we call as transcription is that when DNA is replicated, we say the replication occurs for the entire genome. Okay, so in that process, no DNA is left unreplicated. Okay, so if say I am replicating my DNA, I don't replicate chromosome number 1, 2, 3, 4, 5 and leave the rest as is. Okay, that doesn't occur. So when I replicate the entire complement of 23 chromosomes if I am a haploid cell, or 46 if I am a diploid cell, everything gets copied. Okay, nothing is left. Okay, in a sense, this is like preparing for the exam in the correct way, that you read everything that is there in your syllabus for the exam. Okay, while transcription is slightly different. In transcription, what happens is a particular gene, or what you call a segment of DNA, or a group of genes, or an extended segment of DNA are copied at any time, and some portions of the DNA are never quite transcribed. That means they are never used in this process. So this is like studying in a slightly different way for the exam, where you know that there is going to be a choice between 2 or 3 different types of questions. Okay, let us say that okay, in question number 2 of your exam, there is always a choice between DNA recombination and DNA repair. So you might take a chance and say, okay, I am not going to study recombination at all, because I am going to study only repair, because I know that I am going to get a choice. Okay, so that type of approach is kind of what transcription is, where one doesn't what you call as transcribe the entire DNA that is there in the nucleus of the cell, but only transcribe parts of it. That is either a gene or a group of genes. Okay. Now, how do you define a gene? Okay, this is very important. In the olden days, the gene was always defined as a segment of DNA that codes for a protein. Okay, but that definition is now obsolete. Okay, now we define a gene which actually carries some information content, in the sense that it's a segment of DNA that codes for either protein or RNA. Okay, and the reason for this definition changing from the old definition that gene is a segment which codes for a protein is that because now we know that there are certain segments of DNA which when they get transcribed and they give you the RNA, that RNA is actually the final product. That RNA doesn't go again further to give you a protein. Okay. Okay, and therefore the gene now is defined as a segment of DNA that codes either for a protein or for RNA. Okay, and the interesting part is this gene, which actually is a segment of DNA, may be present on either strand of DNA. Okay, so for example, because DNA is double stranded in the same area, where let us say this is a 5' orientation and this is a 3' orientation, vis-a-vis this is 5' and this is 3' in the anti-parallel strand, the gene, which is a segment which codes for a protein or RNA, may be present either on this strand or on this strand, or actually speaking on both strands. Okay, So the definition of gene is now all encompassing in that it can be there on either of the strands, and it can code either for protein or it can code for RNA. Okay. Any questions at this point before we go further?
No, sir.
Okay. So going further, now here is another aspect of differences. I gave you a little bit of the differences between replication and transcription, but here there are some more points. Okay. One, again, as I said, replication occurs for the entire DNA, transcription occurs only of a part of the DNA. Now, in both cases, the template is a DNA. Okay, so in case of replication, both strands act as a template, and the whole genome is copied to give you a duplicate in a sense. But in transcription, either one of the strands is a template at a given moment, and this is read to give you a copy of RNA, and this is usually constrained or limited to a small portion of a genome at a given time. Okay. Now, when DNA replication occurs, the DNA polymerase, which reads the template, which is called as the DNA dependent DNA polymerase, essentially requires a primer before it can do its synthesis. Okay. So that is what these statements are. That the DNA polymerase, which does the replication, requires a primer. It requires all the nucleotide triphosphates, and the product that you get is double-stranded DNA. Okay. And as this is occurring, the bases which are used are the deoxy ATPs, the deoxy TTPs, the deoxy GTPs, deoxy CTP, and during the process of polymerization, proofreading occurs so that there is no error while doing this duplication. So essentially, this is a summary of what you learned when you did replication. Now look at that, and then opposed to that, look at transcription. The transcription is carried out by what are called as RNA polymerases, but the more correct name is a DNA dependent RNA polymerase because they require DNA as a template. Now, the product that you get is a single stranded RNA, but this RNA polymerase is very interesting. It doesn't require a primer. So it doesn't need a strand already there to which it will add. It can actually de novo initiate strand synthesis. Okay, so it doesn't need to add on to something. It can begin. Okay, in replication, remember there was a little primase which put an RNA primer. That small part of replication process is very similar to transcription in a sense. Okay. Now the bases which are used, okay, in RNA, okay, synthesis are slightly different. Rather than A typically base pairing with T, in RNA you find the base pairing is A with and U. So that means when a template has a, the RNA made incorporates a U. While if the template had a T, that is a DNA template, RNA will incorporate a, and GC is the same. And this is the interesting part, there is no proofreading. Okay, so when RNA is made and later on it is going to be read to give you a protein, there is no mechanism for correcting any errors when the RNA polymerase reads the DNA strand and makes a single stranded RNA. And if there are mistakes in this process, they are just left as is. Okay. Now, before I go further, anybody has an opinion why proofreading is important here, but maybe it is not very important when you make RNA. Okay. Now realize, okay, if I make a wrong RNA and I don't correct it, the protein will be wrong. Okay, and protein may be non-functional. Okay, so given that, why is that we have evolved not to have any proofreading here? Okay, anyone?
So there is post-transcriptional modifications in RNA.
But post-transcriptional modifications have no error correcting function. Okay, none of those post-transcriptional modifications involve correcting an error in the RNA. They involve other processes. Okay, like for example tailing and capping. But if there is an error in the RNA, your PTM, as it is called, post-transcriptional modification, they do not correct the error. Okay, so you will still get a faulty protein if that error stays there. Okay, or you
Or
potentially,
sir, it
yeah.
might be like the
Like,
probability is very less as compared to that happening in DNA.
Uh, and who's this? Who answered this?
Shivam sir.
Okay, so Shivam, see the probability is actually no different. Okay, in fact, DNA dependent RNA polymerase is known to have less fidelity than DNA dependent DNA polymerase. Okay, so in a sense, actually speaking, the chances of making an error are more when RNA polymerase is working as opposed to DNA polymerase. But even then, we do not have any system for proofreading or correcting. So, anyone think why this is so?
So maybe errors in replication may lead to permanent changes
Hmm.
uh, in the DNA itself. So maybe.
And continue the sentence. No, no, you
Uh,
said only one part of the answer. Why you need proofreading for DNA? Okay, and you said see errors there have a issue, and they will it will be permanent. So now you have to answer also the other part. Okay, so as you say the answer, you have to continue into the RNA part also. So continue your
But
answer.
protein thing can be corrected like we have seen gene therapy and all. There is a need to
Ah,
say that in
okay. No, no. See, I understand what you're saying, but remember that gene therapy and all is because that person already has a mutation in the DNA which is permanent. Okay, I'm not talking that here. I'm only talking here that the DNA may be normal, but when I'm doing the transcription to get RNA, the RNA has a mistake. It is not a mistake in the original DNA. Okay, the mistake is in the RNA that is being made. Okay, and still I do not do anything to correct that.
sir, maybe on the later stages, introns are removed, so the faulty genes, whichever is a part of it, could be removed at that stage.
But there is nothing to say that the error will occur only where the intron is. No? Error can occur anywhere.
Yes, sir.
Okay, bolo.
So maybe as the central dogma DNA to RNA, RNA to protein.
Uh-huh.
So initial stage may if we try to remove all the errors
Mhm.
comparatively it will be better. So maybe
Okay, good, good. Okay, now think of it this way. As Sakshi was the one who said no, about DNA
yes.
damage being more dangerous. See, if I have any damage to DNA when I'm replicating my DNA, that DNA is invariably going to be inherited. Okay, it will be passed on to the daughter cell. So any error I make there can have very grave consequences in the daughter cells and maybe later on, if it's in the germ cell, in the progeny itself. Okay, so I do not want errors which occur at random during replication to be part of my DNA. Recombination that occurs is a very controlled way of including changes in the DNA for diversity purposes. It's not an error in the replication. Okay? Now look at what happens during transcription. I'm reading the DNA to get RNA, and I might read this DNA 100 times to give me 100 molecules of RNA. When I do that, 99 molecules of RNA may be right, but one molecule of RNA may have a mistake. So think of it, the remaining 99 RNAs, when they are read to give you the protein, you'll get the correct protein. This one RNA which has a mistake will give you a faulty protein. But these are not permanent things. The RNA which is made and the protein which is made are later degraded. They are not going to be passed on to the progeny as an inheritable trait. Okay, and therefore if I spend too much time correcting the RNA, knowing the fact that I'm going to make multiple copies of RNA and that is going to give multiple copies of protein, I'm just going to waste my time. Okay, waste a lot of energy trying to repair this damage. Okay, now since error is not occurring at a very high rate, okay, when I say error does occur at a higher rate than DNA polymerase, I only mean compared to DNA polymerase, errors are more. But they are not occurring every other base addition. No? It is still 1 in 1 million and 1 in 10 million. So most of the time the RNA made is correct. Once in a while I make a mistake, but since I make many copies of RNA, and then RNA is not something which is going to be permanently in the cell. After a while, when the protein is made, RNA is degraded. Then when you need protein again, you make RNA again. And because of that, I don't need to worry about errors occurring while making RNA, because that error is limited to only 1 or 2 molecules of RNA, and they also are going to be destroyed later. So it's not going to be a permanent affair. Okay, does that make sense to you? Does everyone understand why I do not need to
Yes, sir.
proceed? Because it is not something which is permanent. It is just a very temporary thing. Out of 100 molecules, 1 RNA might have an error, and that again, when it is made into a protein, might give me 1 molecule of protein which is faulty, but remaining 99 molecules of protein are functioning correctly. So I will never see the effects of those minor errors, okay, being very grave. Okay, everyone? Okay, everyone
Yes, sir.
comfortable with what I just said? Okay, so going further, let us look at some of the features of transcription based on you already have a decent understanding of what is going to happen. Okay, so one of the first features is that the transcription is very selective. Okay, when I mean very selective, is that specific sequences, okay, on the DNA mark where the transcription will start and where the transcription will end. Okay, so there are signals which which are given out, which tells your machinery which does the transcription, which is your RNA polymerase and all the other factors that are required to know where to start the transcription process and where to end the transcription process. Okay, and that starting and end will give you a product which is either a RNA or a product which is RNA now, which will later be converted to a protein. Okay, so these signals are the instructions for the enzyme which does the transcription. Okay, now not only that, in addition to where to start and where to stop, these signals or these sequences or these factors which are associated with these sequences also tell, in a sense, when to do this and how often to do this. Okay, so there are sequences which are present on the DNA which not only allow you to start and stop correctly, but also regulate how many times this is going to happen and how often this is going to happen. So in a sense, you know how much RNA you are going to get and how often you are going to get that. Okay, so that is one very interesting feature of transcription. Okay, so which also tells you that because you start at some point and end at some point along the DNA, there might be several such start and stop points, and these start and stop are what we call as genes. Okay, because this is what is going to be coding for something, either RNA, a final product, or RNA which will later on become a protein. Okay, so all along your chromosomes, as far as you are concerned, you'll have regions where such sequences are present which allow regulation of what is going to happen, and then looking at that, your polymerase which does RNA synthesis will come, start synthesis at one point, go along and then stop at a certain point to give you what is called as a transcribed product which we call as RNA. Okay, so that is the first feature of transcription. Okay, the second is many of the RNA transcripts that are made, okay, and this is with respect to especially eukaryotic systems, okay, and it is not that it doesn't happen in prokaryotic system, but it occurs to a greater extent in a eukaryotic system. In that the RNA which is made is many times not ready to do its function as is, and it has to be, in a sense, acted upon further to give you what is called as the secondary or the tertiary transcript. Okay, so what you get are referred to as primary transcripts, which, as some person said a few moments ago, no, have to be subject to post-transcriptional modification. Okay, who said that word, post-transcriptional modification? I think it was Rehan.
Sir, me, Ankit.
Ankit. So Ankit, as you said correctly, many of the RNA which is initially produced by the process of transcription is a precursor which undergoes post-transcriptional modification and what is also called as trimming and converted to what is called as a final functional RNA. Okay, so these initial products are called as primary transcripts and the final products are referred to as either the mature RNA or the final functional RNA. Okay. Now, where does all this occur? Okay, all of the transcription, especially in prokaryotes, occurs in the cytoplasm. Okay, and the reason is that these entities do not have a nucleus. So their DNA is present in the cytoplasm itself, and the conversion of DNA to RNA also occurs in the cytoplasm. However, in eukaryotes, which have nuclei and other what you call as cellular organelles, transcription can occur both in the nucleus and in the mitochondria. Okay, it doesn't occur in the cytoplasm. Okay, and again, further within the nucleus, RNA production of the small r kind, what is called as ribosomal RNA production, occurs in the nucleolus, while the production of tRNA and mRNA, which are called as transfer RNA and messenger RNA, they occur in the nucleoplasm. Okay, so what is nucleolus and nucleoplasm? I've defined it here. Okay, so the nucleus stores chromatin, DNA plus protein in a gel-like substance called a nucleoplasm. Okay, and in that gel-like nucleoplasm, that you have transcription to give you tRNA and mRNA. Okay, the nucleolus itself is a condensed region of chromatin where ribosome synthesis occurs, as it correctly said. Well, that's why we say rRNA synthesis occurs in the nucleolus. Okay, so what is the first feature? One, highly selective. Second, it occurs at specific location, and what you get initially are many cases primary transcripts which have to be modified or trimmed to give you the functional RNA. Okay, by what is called as post-translational modification. Okay, now the other part which I didn't talk about in terms of selectivity is that every cell in your body, okay, can potentially have this levels of selectivity which is very unique to that cell. Okay, for example, all the cells of our body contain the same sequence of DNA because they all have evolved from that initial union of the sperm and the ovum, what you call as the embryo or the zygote. Okay, in spite of that, you will find that certain tissues read only certain parts of the DNA. For example, the beta cells of the pancreas are the only cells in your body which read that part of the DNA which is involved in production of RNA which will later on be converted to insulin. Okay, in no other tissue does this occur. Okay, likewise, that gene which when it's converted to RNA and later on to protein gives you growth hormone, only the cells of your pituitary gland read that information. Okay. Similarly, other cells which are involved in other functions might be reading very specific parts of the DNA and only using that information for their function. Okay, so that is also a level of selectivity. Okay, in the sense that not only that there are specific sequences that mark the beginning and the end of the segment, that is one aspect of selectivity. The other aspect is different organs, different cells and different tissues might use different levels of information which is present in the DNA. Okay, so it's like almost like the way you go in the library, right? When you enter the library, you don't go to the library to read everything. Okay, there are millions of books there, thousands of books there. If you still go there with the intention, I'm going to just read everything one after the other, it will not work. many times you go to the library in a very focused fashion. Okay, your teacher must have told you, "Hey, here is an assignment. Why don't you find out, you know, all the compounds, which have a tertiary nitrogen." Okay, so you go there and you might look specifically for that information, and that information is present only in certain books. So you only go to those books, take out those books, look at that information, and then maybe put it back. Okay. Some other person who's sitting in another class must have another assignment. They are interested in, let us say, the side effects of opioid analgesics. So what they will do, they'll go to a collage book, open that collage book, look at the chapter which has got opioids, and there a specific paragraph which has got side effects. So it's a very focused way you are getting the information out from the library. Okay, and something similar is happening in your body. Your DNA or your genome is like your library. Okay, or you can say it is library plus Google plus Wikipedia plus PubMed, everything put together. All information is there, but you use that DNA in a very selective fashion and read only parts of it which are of interest to you as a person, and in this case as a cell. Okay, and that is another degree of selectivity which is a feature of transcription. Okay, does that make sense, everyone? So one is that certain parts of DNA are read very specifically to give RNA. Second, different tissues, different cells read different parts of the DNA, and depending on what functionality they have to perform. And the second is, much of the RNA which is produced is not immediately ready for use. It has to be modified or trimmed before it becomes functional. Okay, everyone? Okay. Any questions at this point?
Yes, sir.
No,
Okay.
sir.
Okay. Yes, sir. In terms of I can go forward, and no, sir, in terms of no questions. Okay. Okay. So as you go further, before you even delve deeply into transcription, here are some things which we had to be clear about. What is the difference between DNA and RNA? Okay, we have looked at a lot about DNA, maybe a little bit of RNA also needs to be looked at now. What is the difference between what we call as DNA polymerases and RNA polymerases? Okay, one which do replication and one which do what is called as transcription. Okay, I will look a little more into E. coli RNA polymerase and its function, because this is a system which has been studied more than the other systems. Okay, and then we look at things like what are the sequences which make up what we call as a promoter. Okay, in a little while I'll tell you what these promoters are. Okay, these are those sequences which you referred to, no? Specific sequences mark the beginning, and specific sequences tell you when to start and how often to start. Part of that is what is called as a promoter. Okay, and what are the actual steps by which transcription occurs? Basically what we call as initiation, elongation, and then how does termination occur? And there are two ways termination occurs, which we'll be looking at a little later. Okay, so yeah.
So that co-promoters and all is the same thing, right? The
Yeah,
prokaryotic promoter.
there is things like promoters, co-promoters, enhancers. Okay.
Co-repressors and all.
Yeah,
So like
several
now
terms.
boxes and all those.
Exactly. All these Pribnov box, Tata box, okay, here, things which you have learned in your 11th and 12th. All those are sequences which are actually what are called as regulatory sequences, which are what tells you where to start, how often to start, when to start, and all those things. Okay. So those are exactly what we mean by kind of a general term, what you call as a promoter, but I should have been more specific, talk about promoter, co-promoter, repressor, co-repressor, and so on and so forth. Okay, so here is a little look at DNA and RNA from the chemistry viewpoint. Okay, so the first difference is, I look at it this way: they have a similar strand structure in that they tend to have a sugar phosphate backbone, and they also have what is called as a polarity, what we call as a 5' end and the 3' end. If you look at both DNA and RNA, you'll find that one end, the 5' carbon which is present in the sugar, will typically have a phosphate, while as you go along the sugar phosphate backbone, the last sugar, okay, to which nothing is attached, tends to have a OH group, what we call as a 3' OH. Okay. Now the second part is in DNA, there is a OH which is present at the 2' position of the sugar. Okay, and that is why it is called as a ribose entity, and therefore the term ribonucleic acid. In contrast, DNA does not have a OH group at the 2' position. It's a deoxy sugar, or more correctly, a deoxyribose sugar which is present in DNA, and therefore the full form of DNA is deoxyribonucleic acid. Okay. Now, the interesting part is the presence of an OH group at this 2' position actually makes RNA very unstable. Okay. And the reason for this instability is that this lone pair which is present on this oxygen can actually attack this phosphate here, and when it attacks the phosphate, it can eliminate this CH2O, which will leave as CH2O. So essentially what will happen is that if left as is, spontaneously RNA simply breaks down into smaller and smaller pieces because of this attack of this OH on the 3' phosphate which is present near to it. And since this is a decent leaving group, it just forms a cyclic structure, what is called as a cyclic 2', 3' nucleotide. Remember when we studied DNA and RNA, you must have heard of cyclic AMP, cyclic GMP, cyclic 3', 5'.
Yes, sir.
This is a similar structure. It's a 2', 3' cyclic phosphate that you will form, but as you form that, your RNA will break here, okay, and the strand will still run away. Then in that strand, some other place this can occur and will break and will leave away. Now this reaction is not possible in DNA, okay, and that is probably one of the reasons why we say that the big evolutionary step was when organisms decided that rather than having RNA as a genetic material, they should have DNA as a genetic material, which has inherently more chemical stability. Okay. Now the other big aspect is that the bases which are present in RNA typically, okay, and I use the word typically because there are exceptions to this rule, typically are adenine and guanine as far as the purines are concerned, and cytosine and uracil as far as pyrimidines are concerned. Unlike this, DNA tends to have adenine and guanine as far as the purines are concerned, but it tends to have cytosine with thymine as far as pyrimidines are concerned. Okay, so again I use the word generally, okay, which means in DNA most of the time it is ATGC, but every once in a while you will find a U. And likewise in RNA, most of the times it is AUGC, but every once in a while you will find a T also in RNA. Okay. Now this has other consequences because when cytosine, for example, undergoes oxidative deamination, and we remember we saw that reaction when we talked about DNA mutations, okay, as part of our discussions. We said that if cytosine is left as is, it can spontaneously deaminate to give you uracil. Okay. So what will happen in case of RNA is that even though it starts off as being composed of A, C, G and U, spontaneously at many places C will be converted to U. Okay. But since U is also part of the RNA, to detect this and an error is very difficult for the repair enzymes. Okay, if they were to repair RNA. Okay. But in case of DNA, the minute this happens, that is deamination occurs to give you uracil, our repair systems immediately know that uracil is never present in DNA, and they will correct that damage and convert it back to a cysteine. Okay. So the second big step that occurred as far as evolution was concerned was to realize that having cytosine as well as uracil in the same genetic material will not be good. Okay, so the big change was where instead of uracil, the organism decided to have thymine as the other pyrimidine. Okay, so two big steps occurred as far as evolution was concerned. One, when the decision was taken that I'm going to go from RNA to DNA and have more stability, and the second was that I'm going to use as pyrimidine cytosine and thymine, because it will allow me to maintain my DNA more correctly and more easily than trying to locate damages when I keep RNA as a material where cytosine uracil interconversion, I shouldn't say interconversion, cytosine conversion to uracil can occur spontaneously, not allowing me to correctly correct this damage in an easy way. Okay, everyone comfortable with these aspects
Yes, sir.
of DNA versus
Yes,
RNA?
sir.
Okay. Now going further, the polymer of ribonucleotide, as I said earlier, is held together by 3' 5' phosphodiester bridges, just like DNA is. But the difference is that they tend to be single stranded, okay, rather than double stranded. Because if you look, when we talk about DNA, we always talk about DNA in a cell being always double stranded, anti-parallel, you know, helical structure, which is bDNA. Okay, but we never use those terminologies when we talk about RNA, and the reason is many times RNA is actually single stranded. Okay, but the beauty is that the RNA is the only molecule known to function both as kind of a repository of information, that is, it has got a storage function and transmission function, but it also plays a role in catalysis. Okay, so it's a very unique molecule because it is carrying information about proteins in it. It is said to have storage function and transmission of genetic information function. But RNA is also known to work almost like an enzyme and do catalysis. Okay, that's why it is said to be the only molecule known to have both storage and transmission functions as far as genetic information is concerned, but also as catalytic function. And you must have heard about things called ribozymes and stuff like that by now. So we know RNA can also have catalytic function. Okay. Now, all RNAs except the RNA genomes of certain viruses, okay, are derived from information which is stored permanently in the DNA. Okay, so when we talk about, remember when we said genetic information flow, we said DNA always goes to RNA and then RNA always goes to protein, and most of the time, therefore, all the RNA that you find in an organism has always been derived from the information which is present in the DNA, except for certain viruses which contain RNA as a genome, where you need to do initial reverse transcription before information flow can occur. Okay, is that clear? Now, as far as RNA itself is concerned, there are three classes of RNA. Okay, what we know by now, and most of you have known of this since based on your earlier biology classes, that there is three types of RNA called ribosomal RNA, which is depicted as small r and capital RNA, messenger RNA, which is abbreviated as small m and capital RNA, and transfer RNA, which is abbreviated as small t and then capital RNA. Okay. Now, relatively speaking, ribosomal RNA are typically large or long. They are very abundant and they are component of ribosomes. So they play a role actually in protein synthesis. As opposed to rRNA, mRNA are typically short. They tend to encode proteins. There are multiple types depending on which gene has been read to give the mRNA to give you a specific protein, and they are rarely abundant. So they are produced when needed, and then later on when no protein synthesis occurring, the mRNA is basically destroyed. And tRNA are very small, and they are actually, in a sense, the go-between between mRNA and rRNA. Okay, so they are those what you call as adapter molecules, as they say. Okay, and therefore they are important in translation. Now, These RNAs themselves are produced by reading DNA, okay, by RNA polymers. Okay, so they are the final products of the information which is actually contained in the DNA, and therefore we make the statement that not all genes actually encode proteins. Some genes only encode for RNA. Okay, and those genes which encode for mRNA are those genes which will finally take the mRNA into the protein. Okay, so tRNAs and rRNA coding DNA sequences are genes which do not code for proteins, and those sequences which initially form mRNA, which will later on become a protein, are those which are encoding proteins. Okay. A little more information about these three types of RNA in terms of content. rRNA, as I said, is the most abundant. Almost 60 to 80% of all the RNA which is there in your body is rRNA. Okay. tRNA, as I said, which is a go-between, which reads the codes on mRNA and then transfers the appropriate amino acid during the translation process, make up 10 to 20% of the RNAs in the cell. And mRNA, as I said, are the least abundant, but more importantly, their abundance is very variable. At some points, they can even be more than 20% if there is active protein synthesis occurring, but most of the time they are less than 5%. Okay, and what they do is they transfer the information from the gene to the ribosome, in a sense, so that you can make proteins. Now, in addition to these three, which are the actual major RNAs, you also find that there are many specialized RNA also present in the cell. For example, what you call as microRNA, what is called as catalytic RNA or ribozyme, or what is called as uRNA. And what do these do? These are very special functions. For example, some of the RNAs, okay, messenger RNA we know, transfer RNA we know, and ribosomal. This hnRNA or heteronuclease RNA, it's a precursor for mRNA and other RNA. Small nuclear RNA, they are involved in mRNA processing. Okay, another small nucleolar RNA or SNO RNA, they play a role in processing ribosomal RNA molecules. Then there is small cytoplasmic RNA, they are involved in selection of proteins for export to outside the cell. Okay, then it's transfer messenger RNA. They are present in bacteria and they add small peptide tags to proteins to facilitate degradation. Okay, so there are several different types of RNA, and these are names you'll hear here around. But in addition to that, you'll have other names like SNRNA, snRNA, etc. And again, look at this. Some of them serve catalytic functions. Okay, for example, ribosomal RNA and tRNA processing scRNA, and this SNO and snRNA, they also serve to have catalytic function. Okay, so there are many such RNAs which are present in our body. However, much of the times we always concentrate on these three types: mRNA, tRNA, and rRNA. But if you get into this area and look at it in more depth as part of, say, post-graduation or your PhD, you'll start getting exposed to some very fascinating other types of molecules which have their own role in how your cell functions. Okay, and sometimes they can have catalytic functions or processing functions, etc., etc. Okay, and you will learn about them at that point. Okay, now RNA polymerases, on the other hand, they are the ones which synthesize RNA and they use DNA as a template. So they are called DNA dependent RNA polymerases, and they also do polymerization in the 5' to 3' direction, which means they will be reading the template which is present in the opposite orientation, just like it occurs in DNA replication. If you remember, in DNA replication, the two strands separate and then you make the what you call as the daughter strand, and when you make the daughter strand, you are making it in the 5' to 3' direction, but you are reading the template in the opposite direction. Okay, because the template always will be going in the 3' to 5'. Okay, now to make this polymer, what we need is, rather than deoxy NTPs, we need what is called as regular NTPs, what is called ATP, GTP, CTP, and UTP. Now, the prokaryotic RNA polymerase is made up of multiple units. It contains of 6 subunits, out of which there are 2 identical alpha ones, and then similar but not identical beta, shown here as beta and beta prime, and then a gamma and I should say omega and a sigma factor. Okay, so 2 alpha, beta, beta prime, and what you call as the omega is a core enzyme, and then you add this alpha and you get sigma, and you get the functional holo enzyme. Okay, and that is shown here also. This, what do? They detect the DNA which is to be transcribed. This beta actually catalyzes the polymerization. Beta prime has a function in opening up the DNA, double-stranded DNA, so that you can separate and one can be used as a template. Still, we really do not know the actual function of this omega subunit. And this sigma fellow, which is very, very important, it actually recognizes the initiation sites. Okay, what we call as promoters. Okay, and where they are, and then based on that, it will know where to start the process of preparation of RNA. Okay. Now, what we'll do is we'll probably stop at this point. It's just about 2 o'clock, and then when we meet next time, we'll continue from here, and we'll talk about what you call as the function of RNA polymerase. Okay. So at this point, we are going to stop at knowing what are the different types of RNA and a little bit about what is RNA polymerase. Okay, I'm going to stop sharing and look for if there are any questions. Okay, so let me look at the chat box because I think Aditi and Ankit usually write there. Okay, sir, Ankit ka question. Okay, can there be direct production of proteins from RNA via translation in retrovirus, or is it necessary for RNA to produce DNA first and then follow the process of transcription? Okay, now this is I know what question you are trying to ask. Okay, now when you use the word retrovirus, okay, by definition, because you use the word retro, it means that RNA has to go to DNA, and then from the DNA you will have transcription and translation. But if I were to answer your question based on what is there in your mind, your question is, if I am an RNA virus, can I have RNA directly giving me protein? Yeah, that can occur. Okay, and that actually occurs. For example, your coronavirus, no? Which is a RNA virus, when it enters your cell, can directly be read by the ribosome and give you proteins. Okay, so yeah, you can have direct production of proteins from RNA. Okay. That is possible entirely, and coronavirus is actually an example. Okay, here Aditi, it was just a thought. Why central dogma so accepted in all organisms? What would happen if DNA, RNA was directly converted to protein? Actually, RNA does get directly converted to protein. Okay, and that is how life probably was at one point, where RNA was a genetic material. And then you had enzymes which would copy RNA and also convert RNA to protein. That's how many viruses are. Okay, many of our RNA viruses, especially as you are now feeling the effects of coronavirus, poliovirus, influenza virus, they don't need DNA only. They don't go through the DNA process itself. Later on, when people realized, as evolution occurred, and it was realized that RNA is unstable, there are more chances of damage to your genetic material that can occur, that an evolutionary step occurred where the genetic material, rather than being RNA, was DNA in some of the evolved organisms. Now when that happened, then at that point, because the RNA to protein machinery was already there as part of the evolution, the evolutionary step was simply take DNA and then use that same RNA protein machinery which is there and include a step of transcription. Okay, so it is not something which is accepted in all organisms. It is just that organisms evolved when RNA protein was already there. So when I jump to DNA as a genetic material, rather than thinking that how can I make DNA say directly protein, I kept using the machinery which was already there. Okay, Does that make sense, Aditi? Okay. So it is not the question of exception. It's probably just how evolution occurred. So already some machinery was there. I started using it, but I started using DNA as my material. Okay. So let us say, is there any other question? Okay. Both of them say thank you. Any
Sir,
other question? Anyone? Yeah. Yeah.
sir, I have a question. Like it is again not related to today's topic.
Yeah. Yeah. That's
Uh,
okay.
uh, yeah. So I wanted to ask you why that mitochondrial DNA is more robust as compared to the DNA of the normal cells. Is because of the structure or it's something else?
No, no. Why do you say mitochondrial DNA is more robust? It is not at
So
all
I
more robust. It is same.
don't know because I had heard somewhere like there was a forensic case in which they found a mitochondrial DNA on a body which were like preserved for many years, and so they concluded that.
No, no,
That's why.
no, I think nothing like that. Okay, the thing is that if you look at your own DNA, no, which is present in a nucleus, so it's kind of protected inside the cell. And if you look at mitochondrial nucleus, mitochondrial DNA, which is also present within the mitochondria. Okay, so in a sense, both your DNA have got little bit of protection. They are in a organelle, in a sense. The difference is because you have got more DNA, you have got 3 billion base pairs. Remember, chances of damage occurring to your DNA is more. Okay. While you have mitochondrial DNA, which is much smaller than your genomic DNA, so the chances of damaging occurring to it in terms of probability is very less. So it's almost like if I have two people, standing in front of me, and your class, right, and say you have a person who is very thin and small, and right to next to him there is a person who is much taller and much heftier, and I'm trying to throw a stone. Who has got a higher chance of getting hit by the stone? The bigger person who has got a bigger height and who has got a wider body. Okay. So similarly, if chemicals or oxidations or reactive oxygen species are going to damage the DNA when it is just sitting there, the chances of damage occurring to genomic DNA is much more because it is just more and it has got more bulk than mitochondrial DNA. But other than that, stability wise, both are DNA. One is linear, one is circular. There is no absolute difference in terms of chemical reason for difference in what is called as stability. Okay. So I don't know. Maybe I have to read that paper that where you found it in terms of in what context they said that mitochondrial DNA is got more stability. Okay. It is probably more apparent stability rather than real stability. Okay.
Okay, sir.
Does that
Thank you.
answer your question? Yeah. And if you
Yes,
can
sir.
find that source, let me know. We can read it
Uh,
again and look
yes,
at
I
what
will
they
send you,
say.
sir.
Okay. And we look at it. Okay. Any other question? Any other question? Anyone? Okay. If not, can I stop the recording?
Yes, sir.
Yes.
I'll stop the recording.